Hello again. This is my presentation on penicillin, the first antibiotic, and how it is used to destroy certain microbes in the body, and how some of those microbes have become resistant to penicillin, and how they kind of transfer that information between cells. So to start, we'll give a little background on penicillin. So before penicillin, there was no treatment for that was effective against infections. They had treatments that they were using, but they were very primitive. They didn't see many results, or there were lots of side effects from them. So in September of 1928, Alexander Fleming discovered penicillin from mold that was growing on a petri dish that contained Staphylococcus. This picture over here is kind of an example of what the mold looked like, but this isn't exactly the petri dish that he was using. But the mold was growing in an area, and the surrounding area where the mold juice was visible, that was surrounding the growth, was capable of killing a large range of harmful bacteria like Streptococcus and Staphylococcus, Meningococcus, and Diphtheria bacillus. And the first isolations of penicillin proved very unstable when they tried to separate it and use it towards the activation of actually killing harmful bacteria. But this was not actually utilized until 1939, when Howard Florey, Ernst Chain, and their other colleagues turned penicillin into the life-saving drug that it is today. So the mechanisms of penicillin: it's a group of antibacterial drugs that attack a wide range of bacteria. Penicillium fungi is where the drug penicillin actually arose from. It can be taken orally or through injections. It was the first antibiotic that was used by doctors, and the way it works is by interfering with the cell walls of bacteria. So this is done by the penicillin directly acting upon the peptidoglycans, which provide structure for the cells of the bacteria. Peptidoglycans create a mesh-like structure around the plasma membrane in the bacterial cells. This allows for the strength of the walls to increase and prevents outside materials from entering the cell that can interfere with the bacteria's function and growth. The newly produced peptidoglycans fill the holes that form when the bacteria multiply, so the link that binds the peptidoglycans is then blocked by penicillin, and this prevents the bacteria from closing the holes in its cell walls. Because they have these openings, the bacterium will burst when the surrounding fluid from the outside environment has a water concentration higher than what is inside the bacteria, which causes the water to rush into the cell through the holes, and that is when the cell bursts. So this picture over here, sorry, let me kind of make it a little bit bigger so you can see. This is just showing one of those bacterium cells and how the penicillin activates on different mechanisms within the cell in order to open these holes and kind of destroy the peptidoglycans so that the holes remain open so that the water can rush into the cell and cause it to burst, and that causes the cell to die so that it can't multiply and spread throughout the body anymore. Drug resistance occurs in a lot of bacteria. Once they become used to the drug that has been introduced into the cell, they can actually store this information in their DNA and share it with other cells. So I just have a little quote over here that says, bacteria develop resistance mechanisms by using instructions provided by their DNA. So these resistance genes are often found in plasmids, which are just small pieces of DNA that carry instructions between germs or microbes. This is what allows some of these microbes to share their DNA with one another to help them become resistant. So I have a little picture down here. I'm going to make it bigger so we can see. This is just one example of how antibiotic resistance happens. So for example, if you're taking an antibiotic and you don't take the whole thing, or it's not strong enough to kill all of the bacteria, that's when resistance can occur. So you have all the germs that maybe one or a couple of them are resistant to the drug. The antibiotics that are introduced into the system kill the bacteria that's causing the illness, but they can also kill the good bacteria, which is protecting the body from infection. So the drug-resistant bacteria is now able to grow and take over because all those good bacteria have been eliminated as well, so there's nothing in the body that can fight against infection. And then some of the bacteria give their drug resistance to other bacteria through their DNA, which was that other part that I mentioned earlier. So they have, bacteria has different mechanisms with how they can specifically become resistant. So they have different defense strategies, and this allows them to become resistant. They can restrict access of the antibiotic by changing their entryways or the number of entryways that they have. For example, gram-negative bacteria have membranes that are used for protection from their environment. This membrane is basically a selectively permeable membrane that can keep antibiotics from entering. They can get rid of antibiotics by removing it from the cell using their pumps, so they can pump it out of the cell when it enters. They can change or destroy the antibiotics by using enzymes that break down the drug and then remove it from the cell, or they can bypass the effects of the antibiotic by developing new processes that avoid using the antibiotic's target. So if there's like a target marker in the cell that the antibiotic is made for, they can block it by using different targets within the cell and kind of deactivate the old ones that the antibiotic was attaching to. They can also change the targets of the antibiotic so that it can no longer fit in the desired target of the cell. And then just another little area of drug resistance. This picture is actually showing, let me make it bigger. So this shows those mechanisms of how those antibiotic or the bacteria become resistant. So here is the target that's being modified so that so the circle obviously can't fit in this like triangular shape, so it can no longer attach there. The wall is being changed right here of the cell, so the antibiotics are basically bouncing off of it because they have no way to enter the cell and interact with it and destroy it. These are the little pumps that they can change, so they can have activation of drug efflux pumps, which is basically pushing it out as soon as it comes in. It just pushes it right back out of the cell. And then we have inactivation of the drug by the enzyme. So we have the enzyme right here that has attached itself to the drug and is going to inactivate it. And then this is just the bacteria with the cell wall and the cytoplasm and the little plasmid that carries the DNA from earlier that I mentioned. So because of all these resistance mechanisms that it has, it can now be transferred into this plasmid, which controls the DNA, and be shared with other bacteria as it multiplies. And now we're going to talk a little bit about more penicillin and how it works more at a molecular level. So it kills the bacteria by binding to the beta-lactam ring to the DD transpeptidase, which inhibits cross-linking activity and prevents a new cell from forming. So this also makes the bacteria more vulnerable, so because it no longer has a cell wall and it's exposed to the water and the molecular pressures from the outside environment. And exposure to these causes the cell to burst from the water, and it causes the cell to die. So this is a picture of what is happening to one of those cells. So you can see this is the cell here, and this is what happens when all of that pressure comes in. So the cell has burst and is now kind of leaking out into this is probably like a microscope that they're looking under, so it's leaking out onto that area. And over here, like last time, we just have the molecular structure of penicillin, and this is the Chimera program that I'm using to let you see what this looks like. So you can just kind of zoom in and see the structure of penicillin. You can remove the ribbons, so that way you can kind of zoom in and actually see a better idea of the structure, just less bound by those ribbons. And then we can put them back. You can just kind of see how like complex it is and how all of these little areas connect together to form this molecule. And then on the last page of my presentation, I just have the articles and websites that I looked up to find this information. So I hope you found this helpful, and I hope it kind of broadens your knowledge on how penicillin works and how it destroys cells so that they can no longer make an illness in the body. Thank you.